Good morning, everyone, and welcome to Liverpool. My name is Dr. Laura Ajram. I'm the Chief Executive of the British Neuroscience Association, and it is my absolute pleasure to welcome you here to the 7th International Festival of Neuroscience, hosted by the British Neuroscience Association. Over the next 3 days, we'll be joined by over 1,100 neuroscientists from across the world, all here to share the latest in neuroscience research. We believe our strength at the BNA is in the diversity of our membership, and we hope to reflect that diversity in the breadth of the scientific programme that we've arranged for you over the next couple of days. From fundamental and computational neuroscience to drug discovery and clinical practice, early career researchers to esteemed professors, we have a whole breadth of our community here with us this week. And we cover that breadth of neuroscience sectors, career stages, and disciplines because we believe that by bringing them together in one place, we can share, learn, and advance neuroscience together. We've also got a fantastic social event programme planned for you as well. It was lovely to see so many of you at the welcome reception last night, and hopefully we'll see many more of you at our social event tonight. If you haven't got your tickets, you can buy them down at the reception desk. And we've also got today a really special event in partnership with the Queen's Reading Room, so I really do encourage you all to stay with us tonight for that public lecture at 6:30 back here in Hall 1. Before we start, I want to say a huge thank you to our sponsors, our partners, and our exhibitors, without whom this festival would not be possible. Please do spend your time down in Hall 2 and speak to all of our exhibitors, all of our people there that have come with their stands to share how they are supporting the neuroscience community. So with that, I hope you all have a wonderful 3 days with us. And it is my pleasure to introduce our first plenary speaker, who will be introduced by Professor Ian Digid from the Simons Initiative from the Developing Brain, who's one of our partners. To introduce the first plenary speaker and to open our session today. So thank you.
Okay, so good morning, everyone, and welcome to this first plenary lecture, the BNA meeting 2025, and hopefully this is going to be an exciting start to what will hopefully be a packed week of stimulating science. So we're going to start proceedings with a plenary lecture that's generously supported by the Simons Initiative for the Developing Brain, and it's my great honour to introduce today's distinguished speaker, Professor Sir Adrian Bird, who's the Buchanan Professor of Genetics at the University of Edinburgh. Now, he's a true visionary scientist whose work has fundamentally transformed our understanding of epigenetics and neurodevelopmental disorders. A graduate of both University of Sussex and Edinburgh, Sir Adrian's career has spanned over 5 decades of groundbreaking discoveries. Now, he's best known for his pioneering research on DNA methylation in CPG islands, which revealed circuit mechanisms of gene regulation. And this work has sort of laid the foundation for the modern field of epigenetics, demonstrating how chemical modifications to DNA can influence cellular function without altering the genetic code itself. Among his many achievements, Sir Adrian's lab identified MECP2, a protein crucial for interpreting DNA methylation signals. Now, this discovery took on profound medical significance when mutations in MECP2 were linked to Rett syndrome, a severe neurological disorder. And in his landmark 2007 study, his team demonstrated that Rett syndrome in mice could be reversed, offering hope for therapeutic interventions and challenging the notion that neurodevelopmental and neuromaintenance conditions are irreversible. So that hope is now actually being realized with the first clinical trials being conducted using gene therapy approaches to treat Rett syndrome. And Adrian's contributions have earned him numerous accolades, to name but a few: the Buchanan Medical of the Royal Society, the Shaw Prize in Life Sciences and Medicine, the Brain Prize, and obviously his knighthood for services to science. So beyond his tremendous achievements, Sir Adrian is a fantastic mentor and colleague who continues to inspire generations of scientists interested in the complexities of the genome and its role in health and disease. So with that, please join me in welcoming Professor Sir Adrian Bird.
Thank you. Thank you very much for that kind introduction. Very kind introduction. And thank you also for showing up at the prime family time of 8:30 on a Monday morning. At least those of you who are Everton or Arsenal supporters will have had an early night. This is slightly different in title from what I submitted because I forgot to change this title, but the talk is the same as I would have given. Yeah, I'm just making sure I understand how to change the slides forward with the other title. I don't have that many talks. So the talk's going to be in three sort of bits. I know it's a good idea to have one seamless narrative all the way through, but I'm going to introduce Rett syndrome and MECP2. It's going to be more about the function and the understanding of it than the use of it for therapy, but I will allude to the therapeutic issues. And then I will talk about two unpublished stories that I hope I can give enough detail in a short time to be able to interest you. So as Ian alluded to, our sort of entry into this area started with an interest in DNA methylation because there is a modification, there is a methyl group, a sort of nutty hydrophobic lump that gets added to DNA after the DNA is synthesized, and it's added in a pattern. It doesn't alter the genetic code. It's mostly, but not exclusively, as we will hear, in the dinucleotide CG, and it's the cytosine that gets methylated. The pattern implies that there is information there. And so we were interested in understanding what that pattern means. And one route to doing that would be to find proteins that could tell the difference between a CG that's got a methyl group on it and a CG that hasn't. CG is self-complementary, so therefore, because the DNA strands go in opposite directions, and so they occur in pairs. So there's CG on both strands. That's why there are two yellow and red blobs in each region of that cartoon. We found this protein, which I gave the cumbersome name of MECP2. People sometimes call it MECP2, which is a lot easier to say, but I've never trained myself to be able to do that. And we found it because we just took DNA that was either methylated or unmethylated and probed a gel which had been transferred to a membrane containing nuclear proteins from, in this case, brain. And what you see is that when we probed with a radioactive methylated piece of DNA, we saw a band. And when we took the same DNA sequence with no methyl groups on it, we didn't see a band. And so this protein was MECP2. And here are the two methyl groups again. I'm afraid the colors are totally inconsistent throughout the talk. But the DNA by A domain in this protein interacts with the methyl groups, and we understand in atomic detail how that works. So our goal, as you can was try to understand what DNA methylation means, and using this angle as a way of finding out what this protein does, hoping that we would find out what DNA methylation does, and focusing on gene expression and the regulation of that. But then the Zogby lab, Huda Zogby's lab, was searching for the genetic underpinning of Rett syndrome, and they found, in a phrase, that almost all Rett syndrome is caused by mutations in the MECP2 gene. Rett syndrome is a sort of a very profound delayed onset disorder. Kids quite often learn to walk and to talk at about 12 to 18 months, and then they hit a wall, and from which they never recover. And the lifespan is 40, 50, 60 years these days, at least if you have available 24-hour nursing. So it's a devastating disorder. Hypomorphic mutations give rise to X-linked intellectual disability, learning disability, but interestingly, duplication of the gene gives a syndrome as bad, actually more fatal than Rett syndrome. So too much of this protein or too little of this protein turn out to be very deleterious for particularly emphasizing neurological functions. So you notice that all of the individuals shown in the previous slide with Rett syndrome were girls, and that's because of the effects of X chromosome inactivation. The gene is on the X chromosome. Like all X-linked disorders, males are more severely affected, and actually males with these mutations don't survive. But a female embryo that acquires the MECP2 a mutated MECP2 gene is therefore heterozygous, and then following random X chromosome inactivation, they either inactivate in some cells the wild type gene, in which case this cell is MECP2 null functionally, or they inactivate the mutant gene, in which case you have a cell which is MECP2 plus and therefore completely wild type. And it's this admixture of, this is one of the alleles is tagged here to show this is the part of the dentate gyrus in a mouse brain, and it just shows that there's a very fine-grained interspersion of cells that have inactivated one or other of the alleles. Males, as I say, don't survive because every cell is null. Females do survive, but at the very significant price of Rett syndrome. So a mouse model, which we made some time ago, Jackie Guy, who will feature quite a lot in this story, she made a mutation, a knockout mutation. So this is now the male. So this is fatal in humans, and it's fatal in mice. Human males are born very often, but have serious issues and die usually during toddlerhood. Females looked absolutely fine, and this has the advantage you can breed these mice on and maintain the mutation in a mouse line. But about between 3 and 9 months, they acquire phenotypes which resemble Rett syndrome. So in other words, this is a pretty good model of the human disorder, fatal in males and in females, delayed onset, giving rise to a chronic disorder that has a normal lifespan. So this involves tremor and motor problems and breathing arrhythmia, all of which are also features of the human disorder. So a few things about MECP2. It's highly abundant and essential in the brain. So what happens is in a few weeks after birth, this is a mouse here, but the same thing happens in humans. The amount in the brain goes up dramatically. And it's actually, this just shows that it's in the brain. MECP2 is expressed everywhere, but it's particularly high in the brain with the possible exception of the cerebellum. And when I say it's particularly highly expressed, I really mean astronomically highly. There's 17 million molecules per nucleus. If you think of a typical transcription factor, it's usually about 10,000 to 100,000 copies. This is several orders of magnitude more abundant than that. In fact, it's nearly as abundant as histones. There's enough of this protein to sort of coat the genome. So it's expressed everywhere. Is it important everywhere? Well, for the phenotypes we recognize in Rett syndrome, the answer is no. It's much more important in the brain than everywhere else. So if you have a wild-type mouse, it's wild-type in phenotype, but not surprisingly. This is fading out, actually, this pointer. If you have a knockout, then it's, you get the knockout phenotype that I've sort of described verbally. I'll show you an image, a film later. If you knock it out only in the brain, then you get the same severe phenotype as if you knock it out everywhere. And if you knock it out everywhere, except in the brain, the mouse is essentially wild type. Not quite, but essentially wild type. So in other words, where MECP2 matters most is in the nervous system. So it's classed as a neurodevelopmental disorder, which is a sort of loose phrase people use. It's sort of, Wikipedia says it's a disorder that affects the development of the nervous system. There's a sort of implication that it's, which I think is, general, that once you have a problem with the development of your brain, then nothing can be done about it subsequently. But we had the opportunity to test this. There is no cell death apparent in either the patients from autopsy brains or in mouse models, and that's because you have the two X's inactivated, and therefore you can look at the ratio between inactivation of one and the other, and there isn't really much change. So the neurons, though, are somewhat simplified and smaller by this sort of cartoon of Scholl analysis, and this begs the question, if you put MECP2 back, what happens? And the expectation, of course, ours and everybody else's, was that nothing would happen. But as Ian alluded to, something does. This is a male with advanced phenotypic features. It doesn't really move. It's got this tremor. There's breathing arrhythmia that you can see if you follow the flank here. The gait is abnormal. And if you pick it up by its tail, it does this hind limb clasping, which is shown here. Typical of, it's not specific for Rett syndrome, this is typical of mice with neurological, quite a broad range of neurological issues. But then this is the same animal after we have reactivated the MECP2 gene that was silent in this mouse, but we were able to activate it, and the mouse is now disgracefully healthy. So, in fact, it lived for over a year and was fertile. So, activation of the gene reverses neurological phenotypes in both males and females. We showed this in females as well. And inactivation of the MECP2 gene, this is something I haven't shown you the evidence for, is actually lethal. So, what this sort of says is that you don't need MECP2 to make a brain, because these animals had virtually no MECP2 at all, and yet when you put MECP2 back when they're old, if you like, well, at least a month old, you restore its function. And the same is true for the females. Animals were a year old and had the phenotype, and you could put it back. So this implies that you don't need MECP2 to make a brain. You just need MECP2 to keep it going once you've got it. This reversibility of this kind, it actually took quite a long time for this to be extended to other animals. This is partly because of other gene mutation, monogenic causes of neurodevelopmental disorders. Most have still not been tested, but nevertheless, there's some reversibility, and it's variable in extent for some of these other genes as well. And this is Jackie Guy, who did these experiments way back and continues to do experiments in the lab. So this has led on, I'm not really going to go into the therapeutic side of this, but it has led on to 2 clinical trials, and Taysha and Neurogene they're underway now. This is a phase 1/2. It uses a mini MECP2, which I'll describe in a moment. Intrathecal administration, adults initially only, but now pediatric as well. And this is the other one. This is Neurogene. They're using full-length MECP2. They're using a different route of administration and pediatric subjects from the start. And there have been press releases. No paper has emerged from any of this. But the words used imply that they are seeing an effect. Obviously, phase 1 is really just safety, but they are seeing an improvement, at least anecdotal, and it's sort of, one would like to see something quantitative, of course. There has been a setback here. Low dose, a report about low dose was full of quite dramatic cases of girls being able to do things that they couldn't do before. But then a high dose produced regrettably, one fatality. And this has cast a shadow over everything, as you might expect. They are now resuming low dose, and I guess what we're waiting to see is if this, does this actually really make a difference, and how big is that difference? And that's not known yet, but apparently both of them are going to say something in June. So I've been talking really more here about the top-down, the pathology and the mouse model, and obviously one wants to make a causal link between, this is true for all of these monogenic disorders. We know the mutations. We want to be able to trace in molecular terms, in pathways from the mutation to the pathology. And that chain is not complete for Rett syndrome either. But I'm going to talk now about the bottom end here, about the molecular side, which is more in what our lab is mainly involved in. There, if you look at mutations with no clinical effect, you can see that this is MECP2 protein here. They're all the way through it. But if you look at the ones causing Rett syndrome, they're non-randomly distributed, and there are two main regions. This is dying, I'm afraid. There are two main regions, the MBD and the NID, we call them. This is the region I showed you the structure of, binding to methylated sites. There it is. And it binds, and I'm not going to be able to go into this, two things. It binds methyl CG, which I introduced at the beginning, but it also binds another modification, which is more or less neuron-specific. It's fascinating. This kind of modification, methylation of C next to A, and usually, but not absolutely always, but usually followed by C again, this is increases at the same time as MECP2 postnatally, and MECP2 binds to this very well. So it's got two targets in neurons, and one of them is not really found in anywhere else. So does MECP2 really just bind to methylated CG? This is an ongoing, periodic, I've been in this business long enough that I know people say, oh, no, it doesn't, and then they say, oh, yes, it does, and then, no, it doesn't again. And this shows CPG islands, which are non-methylated regions, which are all aligned here. And you can see that DNA methylation goes down because they don't have any methylation, but everywhere else does. And this shows MECP2 binding from data sets from 3 different groups about which were mapping where MECP2 was. And it goes down in all of them over the CPG islands. And this shows a really distant, this is 10 million base pairs, that you can see where methyl CA varies. Methyl C varies quite a lot more than methyl CG, which is more or less uniform. And you can see where the methyl CAC is high, then MECP2 is high by CHIP, chromatin immunoprecipitation, and where it's low, MECP2 binding is low. So that's what that domain does mainly. In our view, there's still bubbling up of possibilities that actually the MBD does something else. But in our opinion, the primary thing it does is recognize a methyl CG based on the evidence from us and others. The other domain is this NID, which stands for N-core interaction domain. And this is a structure that we did collaboratively with Atlanta Cook, which shows this region, which is the sticks in the middle, binding to the sort of donut, which is the WD40 domain of a subunit of N-core. N-core is almost as bad a name as MECP2, but it's a big co-repressor machine which inhibits transcription at the DNA level. So now we have then 2 regions which are absolutely essential for MECP2 function, and we have this model here for what this might mean. So this is now, this is MECP2 here. It's rather an ugly drawing, which I did freehand, and then this is a TBL. This is these are the donuts here. There are 4 of them because it tetramerizes this protein. And this sort of sweet potato here is the N-core, and then there are other subunits here. So MECP2 binds to methylated sites on the DNA, and it also binds to this. And if you bind to N-core via this green donut, a mutation here causes Rett syndrome, and a mutation here in MECP2 also causes Rett syndrome. So that's our model, that this is a recruiter. It recruits the co-repressor to methylated sites in the genome. So I'm not going to show you the test. We've tried to attack this hypothesis. That's your job as a scientist, to try and kill your favorite hypothesis. And we thought if we take away everything in this protein, which is 95% identical between humans and mice, except these two domains, it should work if these are the only bits you need. And this is a mouse that's 11 months old that only has inserted into its genome this mini MECP2. Remember I mentioned that one of the gene therapies is using a mini? This is what it's using, this reduced thing, because the virus has a limited capacity, and so it allows you to fit more into the therapeutic virus for gene therapy. Anyway, that's that. So if you put a stop cassette inside this, so this is the way in which we did the reversal. We stopped transcription by putting this thing in that stops gene expression, and then you can wait until the animal gets sick because it has no MECP2 at all, and then switch on this mini gene, and you find that it's activated by tamoxifen. You find that the animals that controls die progressively, but the animals where you've reversed get better, and the symptoms that they had, the phenotypes they had, reverse. So this is capable of supporting many of the functions of a wild-type MECP2, even though it's missing 2/3 of the protein. So what does it all mean for gene expression? I'm not going to go into this in much detail, but if you take away MECP2, initially people looked, and it was disappointing to see how subtle the gene expression changes were. In fact, nobody could detect them when it was microarrays and things like that. But now it's pretty clear that there are genes that have very few MECP2 binding sites, in other words, not much methylation, go up a little, or they're going down here, but actually you don't really know where 0 is. And then genes with a lot of binding sites go up. And the more binding sites you have for MECP2, the more transcription goes up. And just to show that this is a real effect, if you overexpress MECP2, and remember overexpression causes another syndrome, MECP2 duplication syndrome, then these same genes now do the opposite. The more binding sites they have, the more they go down. So the annoying thing is that these are subtle effects. What you want is, 5 genes that are the real targets of this protein, and it could be therapeutic targets as well. But actually what you find is that thousands of genes are affected, but they're not affected very much. So this is the hypothalamus forebrain visual cortex, just showing you that this correlation exists, and these are different labs who have produced RNA-seq to look at the effects of knocking out MECP2. So this is a consistent effect. The R-squareds are not massively impressive, but they are significant. So MECP2 has an effect, a variable effect, depending on how much methylation there is, on genes that is modulatory, if you like. And so the sort of simple explanation for reversibility is that here are the lollipops. The green ones are methyl CG, the red ones are methyl CAC. This is just a cartoon. And MECP2, this one thing here, purple thing, binds, and then NCOR visits. They're not traveling together. Visits is recruited, and then that leads to the modulation, down modulation of gene expression. And if you take away MECP2, now the down modulation due to that has gone away. Gene expression crudely goes up, and if you put back MECP2, it goes back to doing what it did before, because the methylation is all there. What it reads is all there. So you can, that's a sort of simple-minded explanation for reversibility. So the function of MECP2 it's required throughout life, but is dispensable in early brain development. Neurological symptoms due to its absence can be reversed, and there are 2 trials underway. Primary function is to recruit NCOR co-repressors to methylated sites in the genome, and I should say there are multiple opinions in the literature, as there are in many fields. This, I think, is supported by far the strongest evidence, but people might wrestle me to the ground, other researchers in this field. MECP2 restrains transcription of thousands of genes in a DNA methylation density-dependent manner to, and I think this may be the key word, optimize gene expression. It's almost as though this kind of tweaks gene expression based on this DNA methylation signal that gives you a 100% neuron. And without it, you have a 90% neuron, and that's a problem. So this is an example of DNA methylation regulating gene expression, actually. It's not the kind of switching on, switching off that you see in textbooks as an idea, but it is nevertheless a regulatory act. So now I'm going to talk about a specific story that actually does link to that, and it's Jackie Guy again. Why do small deletions in the dispensable C-terminal region of MECP2 cause Rett syndrome? So I've shown you the MBD here, now yet again a different color, and the NID, and these are the exact mutations in the population, and these are the Rett ones causing Rett syndrome. But there is 10% of all Rett syndrome are caused by mutations here, truncations here, nowhere near either of these two domains. In fact, I've already shown you in the mini-gene, we can chop this whole domain off. So you end up with truncation, and that causes Rett syndrome, and the question is why? There are multiple different deletion boundaries. There are 50, and just 1 to 50 examples of each, and there are really dozens and dozens of different boundaries. Jackie made models of 2 of these, called CTD1 and CTD2, and CTD1 behaved in the way that every other model we've ever made has, namely that the mice got sick, and then this is the aggregate phenotypic score, and they died. This is, in humans, towards the milder end of the spectrum, but it's still horrific. And the mice accordingly live somewhat longer than the knockouts, which are shown here. So the explanation comes from CTD1. Let's ignore the green one for the moment. CTD1, there's almost no protein. It's shorter because it's chopped the C-terminus off, but it's also very non-abundant. So then the other mouse, the green mouse, CTD2, it was wild type, essentially. It didn't die, and it didn't get any phenotype. And when Jackie looked at the amount of protein, it had the normal amount of protein. So having no C-terminus, truncated C-terminus, is fine, provided there's enough of the protein. It's actually the cause of Rett syndrome is the loss of the protein. So what's the explanation for this? Jackie did a sort of an investigation of what was going on here. And it turns out the mouse has a slightly different, a few amino acids different in this region. In the case of CTD1, the key toxic terminus is proline, stop. That's introduced by the mutation. And the amount of protein is low. But in the case of the human gene, it's proline, stop. It would be what you got. But in mice, you get serine proline stop, SPX. And that gives you wild-type levels. So there was something about these last 3 amino acids. And without going into detail, the answer is that if you have proline, which is already tricky on the ribosome for translation, there's special ways of getting through it, followed by another proline, followed by a stop, they lead to loss of the protein and degradation of the RNA, sort of nonsense-mediated decay kind of effect. So that was the explanation of that changed that serine, and there are some other experiments on here I won't go into that validate that. So now another problem arose, which is that if you look in the database, discontinued I should now say, of Rett syndrome mutations, regrettably discontinued, that can be pathogenic or benign, or you look in Nomad, you find that there are quite a lot of deletions which look indistinguishable from the ones that cause Rett syndrome, these ones down here, that don't cause any discernible clinical issue. And there are families that represent this, as well as the anonymous. I mean, you don't really know these people don't have Rett syndrome, but many of them are males, and you know, they've volunteered their blood for this. So the implication is that they have no phenotype, and there are familial examples. Both of these lead to C-terminal truncations, but these cause no phenotype, and these cause Rett syndrome. What's going on here? So, armed with her knowledge, what Jackie did is analyze these different cases. In the case of classical Rett syndrome, they all finish with PPX, every single one. There was one which was claimed ended in this sequence here, but gave Rett syndrome. It turns out that if you model that in mice, which we did, that's not true. So that's a mistake. So all the ones that are neutral variants end in SPRTX. So the reason is it depends on which reading frame you go to. If you, obviously, if you're in frame, you have no phenotype. If you're out of frame, then you have the truncation. But if you end in SPRTX, and all of these mutations end in only these two ends, then you basically have wild-type levels of the protein. All you're missing is the C-terminus, which is dispensable. And so that explained that. And I should say we're using this information to design a therapeutic editing strategy. Jackie is actually converting this X, and has successfully done this in culture, to a tryptophan using an ADAR-based editase, and shown that at least in cultured cells, she can get very high levels of editing that mean you can read through here, and the tryptophan, even though it's not supposed to be there, has no deleterious effect. So this is on the horizon, the possibility of a therapy for this whole class. And this, with one guide RNA, you would be able to do all mutations in this class, assuming one gets to the point where editing is something that's therapeutically feasible. So the intrinsically disordered, and I mention that just because there was a phase when people thought that there was some condensate stuff going on here, but we produced a paper saying that we don't believe that. C-terminal domain is apparently dispensable in humans and mice. Deletions within the CTD only cause loss of MECP2 in one of 3 possible reading frames. The other 2 are benign. The sequence proline-proline stop probably interferes with translation, leading to decay of the nascent protein and its mRNA. And we're trying to develop this therapy. And if Jackie were here, she's not here, but if you want to know more, please discuss. So now the last part of my talk, which is gets us away a little bit from MECP2, but it starts with MECP2. This is about a complex called SET3, which is a variant which is essential for full brain function. And it's the work of Matt List and Katie Payton. They've been leading on this. Other people are involved. And Katie is here, or on the way here at the moment, and she will have a poster. So I urge you to look at that. I'll just make the point here that Jenna is also here, and she's presenting a poster that has nothing to do with this, but she was involved in this work, actually. And she's talking about rat models of neurodevelopmental disorders, which is one of Sid-B's specializations. So please go to those posters. The question that we started with here, I've shown you this rather ugly picture here. The question arose this way. Yes, mutations in MECP2 can cause Rett syndrome. Can mutations in this TBL, in the donut, if you like, cause Rett syndrome? So if not, why not? So TBLR1, this is the donut protein, and this WD40 here, this is the barrel-shaped. It's actually more beautiful than a donut. It depends on your viewpoint, of course. But the N-terminus has this. It's actually a sort of coil-like domain, and the C-terminus. Requiescant in parce. That's the end of the pointer. But it would be quite nice to have a pointer, but I don't know if you have one about your person, Ian? No. This is a picture showing neutral polymorphism pathogenic variants, and you can see there are a lot of pathogenic variants. And this is the tetrameric structure Those, the blobs are the donuts, not drawn as donuts, and you can see there's a coil-coil structure, and that's the N-terminus there. Oh.
See it. It's maybe small.
Yeah, so that's the sort of structural information. The whole structure of NCORE is not known, but bits of it are. And then this is the WD40 domain, and there's MECP2 sitting in it. And so these are the two domains of TBLR1. So it's just to introduce the protein. This is the protein that recruits MECP2. And we're asking, do mutations in this cause Rett syndrome? And the short answer is no. You end up with all sorts of different syndromes from mutations in TBLR1. So the implication is that, the simple implication is, yes, MECP2 binds here, but it's not the only thing. There are other reasons why you might expect mutations in TBL to be different from MECP2, but I think that this gave us the clue that there's something different going on here. Small minority of Rett clinical phenotypes implies that TBLR1 has other functionally important partners. What are those partners? Well, this is, for those of you who know volcano plots, this is a plot where two of these mutations, G70D and D370N, as labeled at the top, were just tested by mass spec. You know, what happens to the proteins? What do they? You compare the wild-type protein with the mutant protein, and what's different shows up as far from the base of the volcano. And you can see ANCAR-D11 and SETD5 are two proteins that we've picked up here. Now, we were interested in these because this is from the 100,000 Genome Project. And this is the frequency of mutations in these genes that cause, they call them neurodevelopmental abnormalities. And you can see ANCAR-D11 is just below the small RNA RNUA4 that was discovered by Nicky Whiffen. That's obviously a major player, but ANCAR-D11 is just below that. MECP2 is there in black because we've already talked about it, but SETD5 is also in the top 10, if that's an appropriate phrase to use for the monogenic causes of neurodevelopmental disorders. So these two proteins we were interested in following up. And they both produce a syndrome, whose description is shown there, including intellectual disability with other stuff, facial abnormalities, skeletal abnormalities, etc. And importantly, the two proteins give syndromes that are often confused with each other because they're so similar. Here, for example, is a paper saying that 3 patients with KBG syndrome, which is caused specifically by ANCAR-D11 mutation, actually turn out to have SETD5 mutations. So the two of them are being confused with each other, and indeed what I'm going to show is that they live together. So this is probably the most complicated slide, and I don't really feel I need to go through it. What we did is look at all of those mutations that are shown, that have their descriptions shown there, right the way across from both domains. And what you could, this is MECP2 and SETD5, and you can see that there are mutations in the C-terminal half, that's the WD40 domain, that cause disease that actually disrupt binding to SETD5 and MECP2, rather similarly, in fact, and for reasons that we now understand. And if we did actually the assay, the animation's in the wrong order here. This is the assay that we used for this. One assay was to insert each mutant protein into a cassette within HEK cells, and then do immunoprecipitation from those cells expressing the mutant, having first knocked out TBLR1 in these cells. And the other one is a more in vitro assay where one just looks at the interaction between two parts of the protein. If they interact, then it lights up nanoluciferase, which is two half nanoluciferases come together to make luciferase. So if you now look at ANCAR-D11, so that's MECP2, which looks like SETD5, ANCAR-D11, those N-terminal mutations in the LIS-H and the F-box-like domain, they actually disrupt the binding to ANCAR-D11. And I'm not going to try and put all this together by experiments for reasons of time. Instead, I'm just going to tell you that that's MECP2 plus NCOR. What we now have is another complex, and this complex involves the same as on the left, NCOR, plus ANCAR-D11 and SETD5. And they're all associated in the same complex, and this kind of fits with the fact that the mutations in one and mutations in the other cause similar phenotypes. So the mutations there cause KBG syndrome, mutations in there cause SETD5 syndrome. So we're sort of putting all of this together. The thing we don't know is on the right here how this interacts with chromatin. So we know there are domains and we don't know what they do, but this is a big unknown with this whole thing. But we're in the middle of working on all this, and probably others are as well. So a prediction is that you would expect mutations in ANCAR-D11 would prevent incorporation into this complex. I've shown you mutations in TBL, the donut thing. These mutations here all cause very specifically failure to bind the NCOR. So in other words, these pathogenic mutations, at least the ones at this cluster at the C-terminus, they mess up the interaction with NCOR. So again, showing it's important. In the case of SETD5, the reason why it looks like MECP2 is because the sequence, we identified this sequence in it that binds to TBL, and it's very closely related to the one in MECP2. And if you make a mutation, that patient mutation also disrupts the interaction with NCOR. So we have evidence here that there is a complex, that these two proteins that are commonly mutated in neurodevelopmental disorders are part of that complex. It's the same it's a derivative of the same complex that MECP2 interacts with. And we made a mouse, and that also says that if you can't interact with NCOR, you phenocopy the knockout of SETD5. So it's clearly a key function of SETD5. So SETD5 is the mammalian equivalent, actually, of a yeast complex. And without going through these protein by protein, it turns out that every single member of the yeast complex, which is shown on the right, has a counterpart in this SET3 complex. And in yeast, it's involved in regulating gene expression, particularly of sporulation genes. So this is a kind of mysterious complex. Nobody knows exactly what it does, but whatever it does is important for proper brain development. And it affects gene expression, so this is a knockout of ANCAR-D11 and a knockout of SETD5 in ES cells, and you can see there's a very strong correlation between the genes that go up with ANCAR-D11, genes that go up with SETD5, and those that go down with ANCAR-D11 and down with SETD5. And I'm not going to, for reasons of time, I think I should be drawing to a close now, but this complex looks like it's interesting to understand, and what we think it does is affect gene expression. We have some preliminary evidence that it affects highly expressed genes, and we need to get to the bottom of this and hopefully understand these at the molecular basis of these neurodevelopmental disorders that are not Rett syndrome, but not a million miles from it. So mutations affecting ANCAR-D11 and SETD5 cause overlapping disorders. Both proteins are in a complex with NCOR via contacts with the TBLR1 subunit. The mammalian ANCAR-D11 SETD5 NCOR complex, it resembles the SET3 complex of yeast. Mammalian SET3 complex is involved in regulating gene expression. So I've actually talked about, and I think I've referred to it enough here, this NCOR complex, and it seems that mutations that compromise the function of NCOR and related complexes underlie several of the most frequently occurring monogenic disorders. And so this is an incentive to try to get to the bottom of these in molecular terms. In both cases, it looks like it's modulatory. And I think, if you're missing a switch, a molecular switch, then you're probably inviable. If you don't have a brain, you don't have neurons, you don't have particular cell types, you die. But what these are doing is de-optimizing gene expression, and de-optimization manifests as illness. And so I think that's possibly why a lot of chromatin regulators are involved in these disorders, because actually they help, but they're not utterly essential for viability. So finally, the people who've done this, Matt List on the left and Katie Payton in the last bit, Jackie Guy I've mentioned. The others are, Jenna, as I say, is here and has a poster about rats, and the others have all contributed significantly. And we're grateful to the Simons Initiative for the Developing Brain, but also Rett Syndrome Research Trust Wellcome and the ERC. And these people, we couldn't have done it also without these facilities, which are very good in Edinburgh. Thanks for your attention.
Well, thank you, Adrian, for a wonderful summary of MECP2 and its interacting proteins. I think we've got around about 5-6 minutes for questions, and I believe there will be roving mics going around, so if you'd like to ask Adrian a question, please raise your hand and wave.
Thank you for an excellent talk. I find it remarkable that the mice recover when you put the gene back. So I'm wondering what you think about how that happens. Do the axons get to the right place already, or are they able to completely grow their axons to the right part of the brain? Do you think it's more the dendrites that are sort of affected by this?
I think this, to be honest, this hasn't been addressed except partially. We had a paper where with Stuart Cobb, where we showed that the morphology did actually change. You got an increased thickness of various regions of the brain with Greek names. Sorry, but I would say whether or not, I mean, there is clearly dendritic growth. So is that supposed to be over? Yes. Yes. Well, I think the one thing that hasn't been investigated so carefully about the reversal is the sort of higher cognitive functions. And maybe that would impact that more. And I know Ian is actually looking into that possibility. Everything, I think, because it's just neurons and everything else that they do looks as though it's reversed, that will be reversible too. But there are those who think that that's unlikely to be true. Time will tell.
Hi, it was a really wonderful talk. My question is, how you said with age, the MEC2 protein levels increase, so how that temporally affects the clockwise, that the genes know what is the age of that mouse. And the follow-up question on that, do you think that could have some impact on dementia or something like, as you were saying, in a higher cognitive function?
Can I just, was your question just about MECP2? Yes. Was it?
Yeah.
Yeah.
I mean, I'm a radiologist, so I don't know much about this, but it's very interesting. So you can answer in any way. If
your question is about the amount of MECP2, it's quite low early on. It's almost as though this protein is really for consolidating neuronal function, the transcriptome, when they mature, when they get mature. And I know people argue about what a mature neuron is, but the amount goes up for quite a long time in humans. But people haven't, for sort of obvious technical reasons, followed individual neuronal subtypes to see which one gets where. And the business about 17 million molecules per nucleus is based on an entire brain. So I would say that the data's not really that clear. Regarding dementia, I don't think there's any reason at this point to believe there is involvement of MECP2 MECP2 misexpression in dementia. I don't think there's any reason to believe that. Now, it doesn't mean it's impossible, but I don't know of anything that implicates it.
Any other questions? One over there.
Yes, I feel as though I'm in the headlights here.
Thank you very much for the talk. I was wondering if you think that there could be involvement of other brain cell types in terms of MECP2 and neurodevelopment.
Yeah, there is. Gail Mandel showed that if you knock out MECP2 in astrocytes, there was some phenotypic effect. I think most people's view now is that, yes, there is a bit of an effect in astrocytes, but the really big effect is in neurons. And if you can reverse it in neurons, then the astrocytic effect isn't big enough to cause a big problem. So that's the way I understand it, and I think other people in the field feel that too. But the answer to your question is it probably is a bit of a function in glial cells of one sort or another, but it isn't as important functionally as a defect in neurons.
I think we'll take one final question.
Yeah, what a wonderful talk, thank you. The levels of recovery for, say, MECP2 are clearly quite important given the effects of duplication and also the anecdotal stuff from the trial. So how easy is it for us to hit the right levels in these kind of recovery studies? And do we need to think about dynamics of these, which might be ongoing in the brain?
Yes, I mean, this is a very important point. And the first point I should make is that the fatality was due to viral overload. It was not actually due to the cargo. That's at least what I have heard. I haven't seen any data. But apparently, AAV9, which is the vector of choice at the moment, unfortunately, gives, in a small proportion of the population, leads to an innate immune response that leads to fatality, commonly. And this is true with other AAV9 treatments that are currently ongoing, such as SMA. It's rare, but it happens. When it happens in a clinical trial, it's obviously seen as much more significant. But the other point you're making is that, given that too much is bad, how do you avoid that? You're going in with a blunderbuss, really. It's quite crude gene therapy. You can't determine how many viruses go into a particular neuron. So the way in which this is being done in both those studies is that they have different microRNA-mediated strategies for making sure that, trying to make sure that they don't overexpress in any particular cell. It's not clear how well those work. I think so long as you're dealing with a virus like AAV9, which hits, only 10% or something of neurons, then you probably aren't going to run into a huge problem. And there's quite a big window of improvement. A tiny bit of MECP2 makes a difference. There's evidence from the mice. Even 10% of the wild-type level is better than 0%. So the more you have, the better off you are up to a certain level, and then presumably the opposite happens. But you're absolutely right. It feels unsatisfactory that one is using such a crude method. And I think this is why genome editing is going to, this is what we really want to do, rewrite the genome so the mistake is gone, rather than dump in an unspecified amount of wild-type gene. But I think provided you're low, probably you can get quite a significant therapeutic effect. Though how you guarantee being low, I think, nobody knows enough in the field, in the clinical side, to know exactly where those boundaries are. But it's a perfectly good point.
So in the interest of time, we'll move on. There's just one announcement that if you're an early career researcher and would like to meet and discuss the science with Adrian, then if you go to the BNA Connect stand, which I believe is downstairs or down the escalator, then you can ask Adrian some questions. Otherwise, I think we'll bring this plenary lecture to a close, and please join me again in thanking Sir Adrian for his wonderful talk.